Very good evening, Lakshita. Very good evening, Karunya. And very good evening, Sanjay, my dear child. Yes, every one of you, very good evening. Happy to see you all and your wishes. So, we are going to start our classes live. Hi, Sanjay. Happy to see you. Very good evening. Okay, so you answered also very great. That is correct. Bacteria, because yes, it divides, but it also multiplies. So it does not know max. Yes, sir? Yeah, I'm so fine, Sanjay. How about you? Yes, Karunya. Very good evening. Happy to see your name. Very good evening, Karunya. Yes, long time no see. Yes, me also same. Yes. So as you answered, it is 100% correct, Sanjay. That is very correct. Yes. So let us begin our session. Yeah, I'm fine with all your great hopes and your good wishes. And till your wishes and prayers are there, nothing will happen. And for your also, I'm praying this year you should succeed, my dear child Sanjay. Shall we start our session now? So, we are going to start the topic. Of course, as you guess, it is biological classification. So, children, what is classification? Hello, Taranika. Very happy to see you after a long time. Yeah. So, classification is nothing but your grouping. Okay. So, we are grouping the organism. That is what we call it as classification. Okay. So, here we are going to group the biological organism. That means living organism. And can anybody guess who is the pioneer of classification? Ancient father of classification is whom? Oh, thank you, Tarunika. That was nice to hear. Thank you. I'm always, when I'm with my students, I laugh. Yes, sir. So, yes, Parthasarathi, very good evening. Happy to see you. Yes, it is Aristotle. Excellent, Karunya. That is the correct answer. Yes, Aristotle is the ancient father of classification. Okay, so those days and all, how they classified the organism, based on the utility. Like it may be useful for clothing or shelter, or it may be for the food. Like that, they classified. And of course, the ancient father Aristotle, he classified the organism like two criteria. One is, of course, plante. Plante he classified like herb, shrub, and trees. And another one is animals. Animals he classified anemia. So what is anemia? Hi. Yes, great jokes. Anemia and anemia. So anemia means without blood and anemia with the blood. So he classified in a different criteria the plant and animal. Before him, the classification was like based on the utilities. So like whether it is used for clothing or for shelter or for food. So of course, ancient father Aristotle also classified like herb, shrub and trees, and also animals, anemia. Now, modern father of classification. Who is our modern father? Of course, you can see the picture. Anybody guess his name? Anyone? It is. Yes, Karunya, excellent answer. With blood and without blood is the answer. Of course, very correct answer. Yes, it is Carolus Linnaeus. So Carolus Linnaeus also made two kingdom classification, as you all know. Two kingdom classification he made, and this two kingdom classification involves Plantae and Animalia. Okay, so he grouped everything in this two kingdom. And after that, many other scientists have evolved, like three kingdom classification, four kingdom classification, five, six, like that, many. But two kingdom classification has got many controversial points. They are not ready to accept this two kingdom classification. Why, children? Like, because he has taken everything in two kingdom classification. That is, he has taken the cyanobacteria and mosses, fungi, ferns, gymnosperm, everything, angiosperm, everything in plantae. So the reason he said it is all having cell wall. Cyanobacteria will be having cell wall, fungi will be having cell wall, all plants will be having cell wall. So it was not accepted. Okay, and also unicellular and multicellular placed together. Like as you know, Chlamydomonas and Spirogyra. Chlamydomonas is a unicellular organism algae. Spirogyra is multicellular, everything together. So that was also not merely accepted. And as you know, autotrophic organism or heterotrophic, again there is no differentiation, everything put together. So, because of these reasons, people are not ready. Even if you think of Euglena, it sometimes shows characteristic features of plants when you put in light. When you put in dark, it shows behave like animals. Okay, so how will you place that in a plant or animal? So there was a controversial point again. So two kingdom classification is many of them are not ready to accept. And today live I have planned some 5 to 6 questions. At the end of the chapter, I'll be asking you a question. Whomsoever is answering, I'll be putting their names. Okay. Yes, composition in cell wall, of course, it differ. But those days they didn't go to that molecular level, no, Karunya. It has cell wall. So he put everything in plantae, Linnaeus. Thereafter only, of course, fungi, chitin, bacteria, peptidoglycan, cyanobacteria is having gelatinous sheath, something like that. So he didn't include those things when he was classifying Karunya. So as we discussed, three kingdom, four kingdom, thereafter five kingdom has come. So five kingdom, these are all the things like Monera, Protista, fungi, plantae, animalia. So of course he is R.H. Whitaker. Any of you remember which year he classified this five kingdom? Yes, Parthasarathi. Is it 1969? Or 68? Yes, it is 1969. He made the five kingdom classification. So one is Monera, another one is Protista, fungi, plantae, and animalia. So what are the basis for classification for these five kingdom? Of course, one is nutrition, and then thallus organization, structure of organism, and then cell structure, reproduction. Apart from that, It also by phylogenetic relationship. So what character is not taken by Whitaker? A possible question. Yes. Very good, Parthasarthi. Excellent answer. It is 1969. Excellent answer. And what character is not taken by Whitaker is presence of nuclear membrane. Okay. So if you see even in NCRT, there is a tabular column like this. We have a tabular column, whether it is mono prokaryote or eukaryote and all that. And then whether it is having nuclear membrane, but it is not proposed by R.H. Whitaker. So he didn't mention about nuclear membrane, of course. Okay, so that we should be very clear. Questions may come. Nuclear membrane presence or absent be included later on. It is not proposed by R.H. Whitaker. Okay. Now, based on the nutrition also it is mentioned, autotrophic and all that. Can you tell me, children, how many of the kingdom are saprophytic in nature? Propose or saprophytic? I'm waiting for your answer. Yes. So next week live, all your names will be in the thumbnail. Those who are answering today. Is it 3 or 4? Or it is 2? Okay, Karunya gave her answer. Fine. Actually, the answer will be yes, Parthasarthi. Answer will be 4. Monera will do saprophytic. It is a decomposer. Protista, slime mold is there. It will do saprophytic. And then fungi is a saprophytic organism. In animals, certain organism are saprophytic. Yes, 4. 4 is the correct answer, Karan. Excellent answer by you. It is 4. Monera, Protista, fungi and animals. Except plant, all kingdom will do saprophytic mode of nutrition, that is even mentioned here. Of course, here Protista, you will have the slime mold will do saprophytic. So next we are moving on to Monera, bacteria. Okay, kingdom Monera, which later on even now subdivided into eubacteria and arachidibacteria by Carl Lewis. But as of R.H. Whitaker classification, it is eubacteria. So kingdom Monera, where is the habit of the Monera? Examples for animals, as you know, there are detrivorous animal, even you can trilobites. It's an insect. This trilobites will do decomposition, like they are saprophytic detrivores. Okay, and cockroaches even we can say detrivorous, but not very much compared to the trilobites. Okay, habitat. So I hope I am clear for you, Karunya. Habitat is, yeah, very good, excellent, cosmopolitan in nature. So that means it is found in air, water, soil, everywhere, unless they are sterile in nature. Every places you could able to find the bacterias. Okay, so that is the condition. Of course, they are cosmopolitan. Very correct answer. Excellent answer by Karunya. So here also you can see so much of bacteria are present. And what are the shapes of bacteria we can able to find? What are the shapes of bacteria? So here you can see, yes, very good, Parthasarathi. Everywhere you can find it is cocci, that is spherical shape, rod shape, bacilli, And can you see a vibrio here? Which is comma shape, and spirilium. Yeah. Bacilli, cocci, vibrio, and comma shape. So you can find different kinds of bacteria. Sometimes they will be in combination also. Here you can see two spherical shaped bacteria together. It is called Diplococcus. Okay, it varies. Yeah, Vibrio. Very good, Karanya and Parthasarathi. Excellent answer. 100% you are correct. Others also can answer. Yes, the question will be there once I finish. Monira, let me see who and all is answering. So let us learn the structure of the bacteria. So you can see a bacteria here. The bacteria outermost is having a capsule. Can you see the capsule? So it may be sometimes rigid or it may be sometimes slimy also. So it is capsule. The next layer inner to that you can find cell wall. The cell wall is made up of peptidoglycan. So peptidoglycan is cell wall. Then plasma membrane we can find. But the plasma membrane is not a continuous one. We can find some foldings here and there. And these foldings are referred as mesosomes. Okay. So what is the function of these mesosomes? Mesosomes will do respiration, DNA duplication, and then cell wall formation. So as you know, cell organelles are absent in bacteria. All the functions of the cell organelles are performed by mesosomes only. Yes, Karanya, excellent answer. It is mesosome. Very correct, excellent. You're rocking. And then you can find, yes, Parthasarathi, excellent. It is doing respiration. Once again, Karunya, very good. Yes. You can find the genetic material. Yes, cell wall formation, respiration, DNA synthesis, etc. Now, as we proceed inside, we can find the genetic material, DNA, in bacteria. What is the nature of DNA do we find here? Is it linear or circular? Is it linear or circular DNA in bacteria? Very good. It is circular without stones. Very correct. Excellent answer. Again by Karunya. Yes, it is circular DNA. And without stone proteins, of course. And inner, you cannot find any organelles. Okay. So, no cell organelles are found. No membrane bound organelles we can find it here. And no nuclear membrane. So, DNA as such dispersed in the cytoplasm of the bacteria. But we can find one organ which is 70S ribosome. And if the bacteria is motile bacteria, what is the organ of locomotion? If it is motile, what is the organ of locomotion? Yes, it is flagella, very correct. They are using their flagella for locomotion and this is the flagella. And you can find Pillai at the time of conjugation they'll be using pillai. Plasmid is extra chromosomal circular DNA, which we will be discussing in biotechnology, of course. And then fimbriae, where it will attach with the rock surface or any hard surface, fimbriae will be useful. So this is regarding the structure of bacteria. So structure helpful to attachment in harder surface is fimbriae. So these three things you should be clear. Flagella is for locomotion. Pili is for conjugation. Fimbriae is for getting attached to the rock or any hard material. Now regarding nutrition. So there are 5 kingdoms are there. Out of that 5 kingdom, all type of nutrition we can find in which organism? So you can find the bacteria, autotrophic, of course photosynthetic autotroph, for example cyanobacteria is there, which has chlorophyll. They can prepare their own food. Chemosynthetic autotroph, where we have purple sulfur bacteria or green sulfur bacteria. All these will come. Okay. And heterotrophic, they may be parasitic also. As you know, E. coli bacteria is parasitic in our smaller intestine, and bacteria only showing various nutrition. Protista is also showing, but we do not have chemosynthetic autotroph there. Okay. Saprophytic, of course, bacillus bacteria is there. Hi, Teja. Very good evening. Happy to see you. Your name? Very good evening, Tejashri. Okay, so we were discussing Monira. Please join with us. So we were discussing bacteria. Bacteria is over. Araki bacteria. So what is the difference? They can survive in any harsh climate. So they can survive in any harsh climate. Yes, Shalini. Hi, very good evening. Happy to see your name. We just finished bacteria and we are continuing with Araki bacteria. And it can survive in any harsh climate because of the presence of dash. Is the question. And I promise today, those who are answering at the end of the session, 5 questions, the names will be highlighted in the next session. Let us see who is answering all 5. Very good, Parthasarthi. Excellent answer. It is different cell wall structure. Excellent answer. So the cell wall structure is different from eubacteria, which allow these bacteria to survive in any extreme climatic condition. That is, it may be a volcano, it may be a high temperature area, or it may be a high saline area, or it may be a cow dung also. So it can survive in any harsh climatic condition. So if it survive in very hot temperature, they are called thermophilic bacteria. And if they survive in very saline condition and all, we used to say halophiles. Hypersaline lagoons. Yes, Karunya, very correct. It is thermophile. Excellent answer. And of course, in ruminants, it is called methanogen. Yes, methanogen, very correct. And out of these 3, which one is helpful in preparing biogas? Hi, Shakti Kumar. And what is that? Bacteria helpful in biogas production? Yes, any of you? Which bacteria is helpful in biogas production? You all know the answer. I also know. Yes, it is methanogen. Very correct, Karunya. Methanogen is helpful in. Yes, Parthasarathi, excellent answer from both of you. Yes, it is methanogen where we prepare biogas. And you can find it in the ruminant animal. Of course, the cow dung, you know, they are preparing biogas from this. And then the cell wall structure is different. Why? Because they have a lipid chain on their side walls. So because of the lipid chain, they could able to tolerate the very high climatic condition. So one such kind of bacteria we are going to discuss is cyanobacteria. Cyanobacteria is otherwise commonly called blue-green algae. This blue-green algae is having chlorophyll A pigment, and this chlorophyll A pigment is similar to that of chlorophyll A found in plant, so that they can able to do photosynthetic. So we justified why they are photosynthetic because they have chlorophyll A pigment, and they are found in, of course, aquatic or even terrestrial, and mostly they are blooming on one particular area. So where they bloom, any of you remember where they bloom, children? They bloom on, they found everywhere, if you see aquatic, terrestrial, but they bloom on. Very good, excellent answer, Parthasarthi. They bloom on polluted waters. Okay, it is NCRT line. So they bloom on polluted water and the shape will be filamentous shape. They may be colonial. Okay, so you can see they are covered by a gelatinous filamentous in nature. They are covered by stagnant water euglena, Karunya, and polluted water only this one, blue-green algae. Okay, you can find a gelatinous sheath. So instead of cell wall, they have a gelatinous sheath. That means they are flexible. And you can find the nitrogen fixing cell, and this nitrogen fixing cell is called heterocyst, which is a very common question. So heterocyst is helpful for fixing nitrogen. Can anybody give example for cyanobacteria? Yes, Shalini, Tejo. Yeah, mucilaginous sheath or gelatinous sheath. It is having somewhat flexible one. Okay. Yeah. So you want me to write mucilaginous sheath. And what is that bacteria example? Very good, Karani, Anabina and Nostoc. Yes, Anabina and Nostoc, very good. Parthasarathi as well as Karunya rocking today. Excellent answer by both of you. So as you can see, this is the heterocyst cell. And if you see this heterotrophic bacteria, they perform various functions like they show metabolic diversity, and they are also decomposers. They fix nitrogen. They fix nitrogen in the form of nitrates, nitrites, and ammonia. So nitrates, nitrites and ammonia. So these are the nitrogen fixing forms. Apart from that, they also fix certain other mineral. Do you remember what are those mineral? Yes, it is helpful as well as harmful heterotrophic bacteria. We are discussing beneficial aspects as of now. And if you remember, it fixes other mineral apart from nitrogen, sulfur, phosphorus, and iron. Of course, nitrogen. Yes, nitrogen, phosphorus, iron, and sulfur is the very correct answer. Excellent answer by both of you. Again, you're rocking. Okay, so this is what is fixed. So this is chemosynthetic autotroph. As I told you already, they may use sulfur or any other mineral to prepare their food. Example, as we know, purple sulfur bacteria or thiobacillus. So these bacteria are chemosynthetic bacteria. Now, beneficial aspects, as you know, lactobacilli is helpful in preparing curd, and we are getting many antibiotic, leguminous plant, fixing nitrogen by leguminous bacteria, rhizobium, decomposition, you can able to see here decomposition. Okay, and harmful is also there when we discuss bacteria beneficial. Harmful, of course, you know, it spoils the food. It causes various disease like cholera, Vibrio cholerae, typhoid, Salmonella typhi, tetanus, um, tetani, Clostridium tetani, citrus canker, Xanthomonas citri. So these are the bacteria which causes the diseases. The names is not given in NCERT, so you need not worry. Just the disease, 4 and yes, very good. Cholera and different bacterial disease or viral disease they may give, and they may ask you which among the following are only bacterial disease. Yes, Karunya, excellent. Now, reproduction. It may undergo asexual reproduction and sexual also. Asexual, as you know, binary fission. So, one bacteria, the nucleus may elongate and center it may constrict, and we may get as a result two daughter cells. And sometimes it produces spores. Spores are produced during what season? Yes. Very good, Karunya. Yes, it is binary fission. What you are observing here is binary fission. You can see here how the bacteria multiply. 1 will become 2, 2 become 4, 4 become 8 and so on. Yes, conjugation is sexual and they produce spores at the time of unfavorable condition. Yeah, unfavorable condition it produce. Very good, Parthasarathi and Karunya. It produced 4 at the time of unfavorable condition. So this is asexual reproduction. What you said, conjugation sexually, I am showing it here in the picture. So this is conjugation. You can see, children, where the DNA from one bacteria, can you see? DNA from one bacteria moving to another bacteria. This pathway we call it as pili or conjugation canal. So as the DNA is getting transferred from one bacteria to another, a genetical recombination is happening. So it is, we call it a sexual reproduction also. But this conjugation where the DNA move from one bacteria to another. So this is conjugation. As you can see, pili is formed and DNA from one bacteria donor cell is moving to the recipient cell. So this is also possible in bacteria, such a small prokaryotic organism showing both asexual and sexual reproduction, various types of nutrition. So it is a complex process. So this is mycoplasma, which is yet another prokaryote, smaller than bacteria, and it is anaerobic. So it does not want any oxygen for its survival. No cell wall is persisting, and so it is called pleomorphic, keeping on changing the shape. So it does not have any proper shape for these organisms, and so only it is called jokers of microbiology. As it is keeping on changing, it is called jokers of microbiology. And as you know, this causes disease in both plants as well as animals. Others, those who are listening and understanding the class, please keep answering. Even if it is wrong, I am your teacher. Please do answer. Yes, it is otherwise called PPLO, pleuropneumonia-like organism. It only causes pneumonia actually. So we just finished one kingdom in RH Whittaker classification. I hope you are all ready to answer the 5 questions. Let me see who is answering first. Net issue may be there. But anyway, if you answer, I'll post your names in the next live session. Let us see. Shall we start? Are you all ready? A smallest living organism. This is our first question. In the 5 kingdom classification, which single kingdom out of the following can include blue-green algae, nitrogen-fixing bacteria, methanogen, arachidibacteria? Yes, Karunya. See, those who are listening can even answer, no issue. Yes, Karunya answered A. Others? Yes, Monira, Parthasarathi and Karunya answered. Anyone else would like to answer? Shalini, Teju, Tarunika? Okay, I'll proceed to the second question. A few organisms known to grow and multiply at the temperature 100 to 105 degrees centigrade, they belong to. Is it thermophilic, subaerial fungi or marine archaebacteria or sulfur bacteria or green algae? Yes. Second question. Shall I count 5? Karunya. 4, 3. Okay. Answers have come, but actually arachidibacteria, whether it is marine arachidibacteria or terrestrial arachidibacteria, we don't mind. But they survive in extreme condition. So that is the correct answer. Fine. Now let us proceed to the next one. In prokaryote, the genetic material is. It happens. Don't worry. Keep rocking. You're rocking all the times. So in prokaryotes, the genetic material is dash. Yes, come on. See, others, please type. Even if it is wrong, no issue, my dear children. Answering is important. Yes, it is D. Both of you, circular DNA without stones is the correct answer. Excellent answer by both of you, Parthasarathi and Karunya. What is true for Araki bacteria? They are allophilic, they are photosynthetic, all are fossils. They are oldest living beings. See, நீங்க நிறைய பேர் ஆன்சர் பண்ணாததுனால இந்த குட்டி கதையை நான் உங்களோட ஷேர் பண்றேன். இந்த குட்டி கதை என்ன சொல்ல வருது அப்படின்னு பாத்தீங்கன்னா, ஒரு பஸ் அதுல ஒரு கண்டக்டர் ஒரு சின்ன வயசுக்காரரு சேர்றாரு வேலைக்கு. சரிங்களா? அந்த பஸ்ல வந்து ஒரு பைல்வான் டெய்லி டிராவல் பண்றாரு. இந்த கண்டக்டர் போய் எப்ப டிக்கெட் கேட்டாலும் பைல்வான் சொல்லுவாரு, இல்ல என்னால டிக்கெட்லாம் எடுக்க முடியாது அப்படின்னு சொல்லிடுவாரு. மறுநாள் கேட்பாரு, மறுநாளும் எடுக்க முடியாதுன்னு சொல்லுவாரு. ஏன்னா அவரோட உருவம் பாத்தீங்கன்னா பைல்வான்ற மாதிரி ரொம்ப ஹைட் அண்ட் வெயிட்டா ஜைஜாண்டிக்கா இருப்பாரு. சோ கண்டக்டர் ரொம்ப சின்னவர்ன்றதுனால பயந்துட்டாரு. அப்புறம் கண்டக்டருக்கு பொறுமை தாங்கல. ஒரு நாள் வந்து அவர் ஜிம்முக்கெல்லாம் போயிட்டு, யா குட்டி ஸ்டோரி டைம். அவர் வந்து என்ன பண்ணுவாரு, கண்டக்டர் போயிட்டு ஜிம்முக்கெல்லாம் போய் பாடி எல்லாம் நல்லா பில்டப் பண்ணிட்டு, அவர் என்ன பண்ணுவாரு, கம்மிங் பேக் டு த இது ஜர்னி அந்த டிராவல் பண்ணும்போது டிக்கெட் கேட்பாரு. நான் வந்து டிக்கெட் எடுக்க மாட்டேன் அப்படின்னு பைல்வான் திரும்பியும் அவர்கிட்ட சொல்லுவாரு. ஏன் அப்படின்னு கேக்குறாரு இந்த டைம் செகண்ட். ஏன் அப்படின்னா என்கிட்ட பாஸ் இருக்கு அப்படின்னு அவர் சொல்றாரு. இது ரொம்ப ஃபன்னி ஸ்டோரி தான். ஆனா இதுல என்னன்னா நம்ம பார்க்க வேண்டியது பயந்துகிட்டே இருந்தா ஒரு டைம் அவர் கேட்டே இருக்க முடியாது. அவரை எக்யூப் பண்ணிக்கிட்டாரு. அந்த பயத்தை போக்குறதுக்கான வழி என்னன்னா அவர் ஜிம்முக்கு போனது அண்ட் அந்த ஜிம்ல அவர் பிராக்டிஸ் பண்ணது அவரோட பாடி பில்டிங் இதெல்லாம் தான் அவர் பயத்தை போக்குறதுக்கான ஒரு இதுவா இருந்தது இல்லையா? அப்போ அதே மாதிரி நாமளும் நம்மளை எக்யூப் பண்ணிக்கணும் ஆஸ் வி ஆர் கோயிங் டு ரைட் த நீட் எக்ஸாம். இப்போ உங்களுக்கே தெரியும் நிறைய டைலமா நிறைய டென்ஷன் அதெல்லாம் இருந்தது. பட் வாட் எவர் இட் இஸ் உங்களோட டெஸ்டினேஷன் கிளியரா இருந்ததுன்னா நம்மளோட எக்யூப்ஸ் என்ன, வி ஷுட் ப்ரிப்பேர் தரலி பிராக்டிஸ். சோ இந்த ஒரு குட்டி ஃபைவ் டெஸ்ட்டுக்கே பயப்பட வேண்டாம். ஈவன் இஃப் இட் இஸ் ராங் லெட் இட் கோ. ஒன்னும் பெருசா விஷயம் இல்ல. ஓகேயா? நான் வித் தட் ஸ்டோரி இஃப் யூ ரியலி லைக் ப்ளீஸ் புட் ஆல்வேஸ் லைக் கமெண்ட் அண்ட் லைக். நான் அதுதான் கேட்பேன் உங்ககிட்ட. அண்ட் தீஸ் ஆர் ஓல்டஸ்ட் லிவிங் திங் அப்படின்றது ரொம்ப கரெக்டான ஆன்சர் பார்த்தசாரதி அண்ட் காருண்யா அண்ட் புஷ்பவதி. வெரி கிரேட். So after the story I got one more answer. Very good. Which bacteria are utilized in gobar gas plant? அடுத்த question. கோபார் கேஸ் பிளான்ட்ல எந்த பாக்டீரியாவை நம்ம யூஸ் பண்றோம்? Karunya gave an answer. Okay. Yes, it is methanogen. Excellent. Gobar gas அப்படின்றது ஒண்ணும் கிடையாது. It is biogas. So that was very correct answer. Excellent answer by both of you. So next we have to move on to kingdom protista. So kingdom protista உங்க எல்லாருக்கும் நல்லா தெரியும். இந்த protista வந்து we have 5 major classes இருக்கு. It is yes Gunashri, happy to see your name and that is a correct answer. Great. But இன்னைக்கு கொடுத்த அந்த 5 questionல 4 question கரெக்டா பண்ணுவாங்க. The great and great Karunya Shruti as well as R Parthasarathi. Excellent. Next time liveல உங்க பேர் வர போறது. So I'll write your name in the live session. Great. Great job. Well done. Keep rocking. So kingdom protista. As you know, these are unicellular eukaryotes. And you can find these organisms everywhere. So landலயும் இருக்காங்க, tongueல இருப்பாங்க, and you can find in aquatic area also. And they have a link with all other kingdom. So link எங்கெல்லாம் இருக்கு அப்படின்னு பாத்தீங்கன்னா பிளான்டோட இருக்கு, அனிமலோட இருக்கு, and ஃபங்கையோட இருக்கு. ஓகேயா? அண்ட் சோ இது மாதிரி எல்லா ஆர்கானிசமோடையும் இவங்க வந்து லிங்க்ல இருக்கிறாங்க. And they undergo reproduction both asexually and sexually through the mechanism called cell fusion. Yes, Karunya, fungi, plant and animal எல்லாரோடையும் அவங்க லிங்க்ல இருப்பாங்க. And zygote formation. So நம்ம ஃபர்ஸ்ட் பார்க்க போறது என்ன அப்படின்னு பாத்தீங்கன்னா கிரிசோஃபைட்ஸ். இந்த கிரிசோஃபைட்ஸ்க்கு வேற என்னெல்லாம் நம்ம பேர் வைக்கிறோம்? Any of you? So chrysophytes is otherwise called desmids or diatoms. Okay, or otherwise golden algae. Very good, excellent answer. Desmids or golden algae. Yes, Gunashri, it is by zygote formation and cell fusion. Excellent answer. So இதெல்லாம் அதோட பேரு. நீங்க இங்க பார்க்கலாம் இந்த golden algae அவங்களோட habitat எங்க இருக்கு? Yes, diatoms, desmids, golden algae இவங்களோட habitat எங்க இருக்கு? Yeah, marine and freshwater அவங்க இது